Good afternoon, students. Welcome to yet another live session of Bio 191, General Biology Practical 1. Last week, you people were not many, probably because of the email facility that was not okay. I hope you people received the email I sent out pertaining to this course. Hello? I hope you received my mail.
Yes, ma'am.
Good. And the time remains same every Monday, 12 noon to 1 p.m. is our time. It remains same every week. So last week was the introductory class. I introduced myself as Professor Morenchuku, being your facilitator for Bio 191. Then I explained to us, we looked at, biology practical, introductory lecture on biology practical, where we examine what biology, importance of practical biology, involvement, what you're supposed to be doing in practical biology, biological diagrams. We looked at this, all this, how we draw, how we label, how we name the specimen, and so on and so forth. And I believe you've seen the recorded class lecture. Today, we are going to move a step further.
Man.
Mute yourself, please. So, at this juncture, I'll start sharing my screen to explain to us what we have today. We are going to look at procuring animal and plant materials for laboratory work. We've seen the intro lecture, looked at biology, what biology is all about, practical biology itself, why study practical biology, what it's all about, look at involvement, what is involved, how we draw our diagrams, and so on and so forth. Now, please mute yourself. Olumide, stop disturbing us. I'll start sharing, because we're going to look at procuring animal and plant materials for laboratory work. Let me start sharing my screen. Please stop disturbing us. I will minimize. Our topic once again is procuring animal and plant materials for laboratory work. Learning outcomes. Yes. On completion of this lesson, you'll be able to place the order with a dealer for purchase of specimen and live animals. Collect live animals from their natural environment. Learn how to identify male and female animals. Collect algae, bryophytes, pteridophytes, and gymnosperms. And collect, press, dry mount, label, store, and preserve angiosperm specimen to make a herbarium. Quickly attempt these activities. How can you procure animal materials? Placing order for the specimen and live animals. Please stop disturbing us. Placing order for the specimen and live animals. Specimens are to be procured from the market. In your institution, we have procurement directory that takes charge of this. First and foremost, you normally write 2 letters to be written from your institution. The first is for inviting quotations from dealers, and the second is a letter placing the order. And normally, when quotations are invited from the dealers, the procurement unit will meet and now invite and choose the dealer with most reasonable prices. At the same time, there should be an alternative arrangement in case of disappointment. The collection of aquatic invertebrate sources, material required. We have nylon nets, fine weave of small animals, and coarse weave for large animals. That we have different sizes of nets, fine for small animals and coarse for large animals. Then we have large clean jars or buckets. Then shallow white pans or papers. So collection of insects, we use sweep nets. Sweep it across of different sizes, different shape, as the case may be, we use to collect insects. We see the bag shape one, round one, square one, and half round. Then collection of insects also, we can use a flash torch and aspirator. Time we flash touch the insects with the lights will keep them at a particular place. Then we can still use aspirator. And talking of collection of plants, first we have to look at the bryophytes. We spread the bryophytes from the place of, we scrape the bryophytes from the place of occurrence with the help of a sharp razor, scalpel, or knife and keep them in polythene bags, within which they remain alive to a number of days. Now we keep these bags loosely tied and in damp condition in laboratory. Then wash the soil growing species with ordinary water to remove soil particles and dirt when attached to plants. Then keep the bags under illumination at 0 to 5 degrees centigrade to keep the plants alive for a longer duration. Then we freeze the material, label, and keep it in the core board. Then next we look at the pteridophytes. How do we collect pteridophytes? We collect them from their natural habitats in mature spore producing stage. We collect the plants with or without strobilurin or mature sporophyll in polythene bags loosely tied at the mouth. Then if the material is large, cut them into pieces, face label, and keep them in a core board. Then let's look at angiosperms. How do we collect them? We collect the roots, stem, leaves, male and female gymnosperms of the plant. We cut the material into small pieces of 3 cm, face, label, and keep them in a core board. Can collect the dry fruits and cones of gymnosperms and preserve them as such. Then let's see collection of hair plants for making a herbarium. Don't pick plants haphazardly. Choose only those plants whose flowers are large and whose organs are easily seen. If possible, collect complete plants with flowers and fruits for easy recognition of species of some families. The underground parts, that's the root, bulb, rhizomes, are often interesting. Try to collect them. If the plant is large, take just a branch with leaves, flower, and fruits. Then carrying the specimen, do not mix up or damage selected plants with others. Place the plant quite flat between newspapers and place in a cupboard folder and fasten in a strip or keep the plant vasculum. Keep in the plant vasculum. Attach a label to each plant, stating details of its dwelling place, dates, name of place, flower color, and any other interesting feature. Examining the plant. Identify the name of the plant with the help of your course tutor. In due case of time, you'll be able to name the plant by yourself. You can also keep a plant with some labeling with it, and it could be identified with the help of the flowers. The help of the flowers, make a few sketches to point out the characteristics which help you in identification. See what you have. Plants have been dried under the paper. This is what we have, the dry plants. Drying, put them on a paper and then dry them under the sun. In conclusion, in this lesson, you've learned that you either buy or collect animal materials. Once materials for laboratory work has been acquired, they must either be killed and preserved for studying or kept alive in suitable conditions for the same purpose of being studied. You go to the natural habitat of plants to find them. Most lower plants have to be fixed before storing away in labeled sterilized bottles. For most higher plants, their preservation process is drying and their storage place is the herbarium. Now, try and work on this activity in your own time. Describe how you will collect and the following animal materials: aquatic invertebrates and insects. In the next lesson, we'll examine preparation of temporary slides. Thank you so much for listening. I will stop sharing for us to ask questions. Any question? Please, if you have a question, signify by raising up your hand. Maybe I will. If you have any questions, signify by raising up your hand. Question, Olumide, unmute yourself and ask your question. Olumide, ask your question. Maybe I'll run through lesson 1 PPT.
Good afternoon,
Olumide,
ma'am.
good afternoon.
Yes, ma'am. Thank you so much for the section, ma'am. Really appreciate, ma'am. But ma'am, I have a little bit of issue joining the class. I just gained access not quite long, and I don't know how I can maybe get the materials if I can follow up later on by myself.
The materials are on the study, on the platform, the facilitation platform. Though we didn't start early today anyway, something came up, I was in a meeting, but the materials are there. Even the recorded lecture will also be there. Please, this is every Monday class, 12 to 1. I'll try as much as possible to be starting on time. Something kept me in a meeting today. And we are all expected to be available. Tell other members of your class, if possible, you can form a WhatsApp group where you'll be communicating. Any other question?
Thank you, ma'am.
Any other question? Okay, the turnout of last Monday was very poor. So let me quickly share my screen and run down last week's work. Last week is an introductory lecture anyway. And then I tried explaining to us what is biology. As you all know from your secondary school point of view, that is the study of life. And let us begin to envisage it, that as students, history, literature, social studies, after classes, they study on their own. They go to library and whatever without going to the lab. But for sciences, physics, chemistry, biology, we still go to lab. Reason we are going to see across here. So for last week, we looked at the following learning outcomes. That at the end of this lesson, you'll be able to state why practicals are important in biology, explain how to successfully carry out practical work in biology, say the involvements in practical biology, and make good biological diagrams. Then we look at the importance of practical biology. As I've already said, after studying in class, we go to the lab. Why all these things? First is for better understanding of subject matter. It enables discovery of facts about organisms, enables scientists to approach problems in a logical, orderly manner, and the proper solutions to challenges of man. Quickly go through this activity. What are the involvements in practical biology? Practical biology involves the following. We have field work. Time we go to the field, natural habitats of the organisms, examine them, look at them, how they interact with other organisms, even with the physical environment. Then times we need to collect this specimen, that's the live organisms, take them to the lab, preserve them, and store them. We can use them immediately. After usage, we still store. Or even without usage, we preserve and store. Then there are times we have to prepare specimen for laboratory studies. There are times we have to make sessions and slides. Sessions means cutting open to see the internal structures. Then there are times we have to study the specimen in the lab. And we looked at biological drawing, how we have to be drawing. So when going to the lab, go with sharpened pencils, not just one. Men, it's not that if there's issue with one, you switch over, not wasting your time resharpening and things like that. Then take to observation. You have to have high sense of observation and follow to instruction, whatever instruction you are given, key to it. Then while drawing, you draw with same continuous line, not woolly line. As you can see in this diagram here, the one marked good is correct, and the one marked bad is not right. So be drawing with wavy line. We talk of views. Which part of the specimen are you drawing from? If you are drawing from the front, it is anterior view. If you are drawing from behind, it is posterior view. If you are drawing from top, it is dorsal view. If you are drawing from beneath, it is ventral view. And if you are drawing from the side, it is lateral view. Now let's see the sections. We have two types of section. Can be longitudinal, cutting open from top to bottom. As we see here, longitudinal section of a mango, where it cuts open, this is what you see. Then it can be transverse, cutting across to give you a transverse section. For example, we see here transverse section of a garlic rose. When we talk of magnification, we are looking at the size of your drawing compared with the object. If it's twice, it is times 2. Equation is being represented by times. If it is half, it is times 0.5, and so on and so forth. Then we talk of labeling. How do we label? We label with straight lines, drawn with ruler and pencil, none of them without arrow, because when there's arrow, the meaning changes to direction. They should not overlap, then we label. One side, but where there are many structures to label, can see label on both sides. You can see the diagram of a rat there, labeling was labeled in the right method, while that of Amoeba wrong, because there you can see the guidelines overlapping themselves. And you talk of headings and the title itself. Heading. First, in your writing your heading, you identify the specimen. What is it? Then you talk of the view. Where are you drawing it from? Is it from the left, from the front, back, top, bottom, side? Then magnification. So you can see lateral, and see lateral diagram of a mango times times 2, meaning that you are drawing from the lecturer point of view, 10 times 2. Then there are times for experimentation, when you have to go to the lab, carry out experiments, either to find out facts or to verify already existing facts. Like you go to the lab, you're giving instruction of what to do, take the beaker, like, okay, experiments, there should be a, okay, I'm coming to that. After performing your experiment, following the instruction, getting your report, then you report. Let's see various session of reporting. First, you have your dates, the purpose of the experiment, that's the aim. For example, experiment to show that oxygen is given off during photosynthesis. That's the aim or purpose. Materials will be provided for you. Beaker, green plants, Bunsen burner, and so on and so forth. Then you'll be given directive, distilled water, on what to do. There's also means of distilled water. Mix it with this, put your plant, boil for so length of time, and so on and so forth. When you follow viciously, you get your results. Then, introducing a microscope. A microscope is an instrument that increases the size of the object, magnification. And you'll be using it in this course because you'll be looking at small organisms, specimens that need to be enlarged. Microscope is quite an expensive instrument, should be handled with care. We have the ocular lens through which you view. You have the arm, you have the base with which you carry it. You have the adjustment, both quartz and fine adjustment for focusing right. Then you have different power lenses, 10, 40, 100, depending. You have the stage for keeping your slide. You have the clip for clipping it. Then you have the projection lens, and so on and so forth. Then the microscope should be handled with care. While cleaning the microscope, you use lens tissue to clean the lenses. In conclusion, in this session, we've learned that practical work is an essential component of theoretical study of biology, the basic essentials and techniques in practical biology, the things you need for practical work, the need to be exact, careful and follow instruction during practical work. Then attend this activity in your spare time. Draw and label a compound microscope, outline the functions of NS's label parts. As I said last week, today's lesson was what we had to look at today. Thank you so much. At this point in time, I'll stop sharing for us to ask some questions. Just run through for the benefits of those that did not attend last week. Questions? Please, if you have any questions, signify by raising up your hands. I mustn't fail to say that we'll be having this every Monday, 12 to 1. Always join via the same link. I'll try connecting on time. Then let your other classmates know. In your class for today, as at now, we have about 350 registered members that are supposed to attend this, though normally it used to be over 1,000. Know you that practical has started for the semester. Practical has started, and you are expected, though we started with second semester courses, 191 will be in 2 weeks' time, then 2 weeks for this, 2 weeks for that. I believe you've been registering. We have practical centers, North Central Abuja, Northeast Bauchi, Northwest Kano, Southeast Enugu, South Benin, and Uyo, and Southwest Lagos VI, Lagos Ikorodu, Lagos Surulere, and also Osogbo and Abeokuta. Thank you. I mustn't fail to say that discussion forum 1 has been opened since last week, and you are expected to go there, log in, and contribute to your own quota. Yes. Ben Colley. Yes. Ask your question. Please unmute yourself, Ben, and
Okay.
ask your question.
Good afternoon, ma'am.
Afternoon.
My question is, why labeling? Is it must you label on the right-handed side, or you can label both on the left-handed side?
It's like you are not with me. I said you label on one side, it can be left or right if there are not many structures. But where there are many structures, you can label on both sides of the
Okay,
same diagram. Yes.
okay. My discussion forum is still locked. It's not open yet, but
Are you
so
sure?
yes, ma.
For which geopolitical zone?
North Central, ma.
I will open it immediately after this.
Okay, ma.
Any other question?
For now,
So
no.
we call it a day and see you next Monday. And I expect over 100 of you around next Monday. Thank you. I'm ending for all. Bye. See you next week.